Right, good evening, everybody. How you're doing today? Can you please let me know if you can hear me? All right, great. So are you able to see the PowerPoint on the left and the blackboard on the right side of the screen? All right, great, great. All right, so the last things that we've been discussing was the smooth muscle contraction, if you remember. And the smooth muscle was classified according to whether it acts like a skeletal muscle or if it acts like a cardiac muscle. And if you remember, the two types of smooth muscle, those are going to be differentiated from one another according to how would they respond to the stimulus. If the set of smooth muscle cells are going to be responding as one unit, we call those are the single unit smooth muscles. Another name for it is the visceral smooth muscles. Remember, this is the type of smooth muscles that we're gonna have in the wall of the gastrointestinal tract and other visceral organs. That's why we call them visceral smooth muscles. If you remember, in order for you to contract a certain segment of your gastrointestinal tract, as an example, you can't just contract part and not contract another part in the same time. So if you're going to be deciding to contract this segment of the gastrointestinal tract, it is important to contract the next portion and the next portion and the next portion. So here, all the smooth muscles on the wall of the this part of the gastrointestinal tract, let's say smooth muscle, let's say small intestine, all of them gonna need to get the stimulus and respond to the stimulus freely. So I need the stimulus to be able to travel freely between those smooth muscle cells for all of them to act as one unit. So we call this as a single unit smooth muscle or visceral smooth muscle. We've seen another type of smooth muscles that will be performing a graded contraction. And if you remember, a graded contraction means it's not essential for you to contract all the muscle cells within a special organ. You're gonna be only recruiting few of them according to your needs. And this is how the skeletal muscles were acting. The same way like the skeletal muscles, we have a type of smooth muscles that performs a graded muscle contraction. What do we call the smooth muscles that will be performing graded muscle contraction? Those are my multi-unit smooth muscles. It's not essential for me to contract every single smooth muscle cell within any specific organ. You're going to be contracting the number of muscle cells that will be satisfying your need. Like what examples that we've discussed last time? Like, for example, if we're looking at the iris, remember, at the center of the iris, we've got this hole through which the light is going to be traveling to the inside of the eye. remember, you're going to be either contracting or dilating the pupil of the eye to regulate the amount of light that will be traveling to the inside of your eye. So it's not an all or none contraction. I have different grades of the contraction that will be regulating the amount of light. So it's not either you fully dilate or you fully constrict the pupil. So it's not either like this or like this. You're gonna have many levels of the contraction in between that you might be in need of. So it's not an all or none contraction like the visceral smooth muscles. Multi-unit will have different grades of contraction. Same concept when we are looking at the blood vessels. In order for me to regulate my blood pressure, I don't have to fully constrict or fully dilate my blood vessels. I need to perform different levels of the contraction in order to regulate the blood pressure according to my needs. So it's not an all or none contraction, it's a graded muscle contraction. Same concept for the bronchi that will be regulating the airflow to your lungs. It's not either you fully constrict the bronchi or you fully dilate the bronchi. You have different stages of contraction of the bronchi to be able to regulate the amount of air that you're allowing in your lungs. So this is the difference on here between a multi-unit smooth muscle and a single unit smooth muscle. We stopped at this point last time, if you remember. Today we are starting our discussion with the homeostatic imbalances that are related to the muscular system. First one that we're looking at today, it's the Duchenne muscle dystrophy. It is one of the most common and severe types of muscular disorders. It's a genetic problem. The defective gene is going to be located on the X chromosome. If you remember back from your general biology class, a female has 2 X chromosomes and the male only have how many X chromosomes? Do males have X chromosomes or not? What do you think? One X chromosome. And the other sex determining chromosome that the males will have is going to be the Y chromosome. So any male will have XY and any female will have XX. General concept, the X chromosome carry a lot of genes other than sex determining genes. Very important genes are going to be located on the X chromosome. Compared to the Y chromosome, it's a much smaller chromosome. It mainly contains the sex determining genes, which allows a person to differentiate into a male. All right, so looking here, females will have XX, males will have XY. So this means what? This means that any male is going to be producing some sperms that will carry the X chromosome and some sperms that will carry the Y chromosome. All females will be producing eggs that only contain X chromosome. So the next population, they have a 50-50 chance of either getting a female or getting a male. Any problem so far?
Hi, boo
Any
boo.
problem?
Something stinks in here. Hi. Smells like trash or garbage. Smells cheesy.
Can you hear me? All right, so again, what we're looking at in here, we're looking at the distribution XX and XY. XX being a female, XY will be a male. So any two, they have a 50% chance of getting a female child and a 50% chance of getting a male child. Right? Is there any problem here? All right. So if I'm telling you that the Duchenne muscle dystrophy is a sex-linked disease, it's an X-linked disease. So the defective gene is going to be present on the X chromosome. If a female has a defective gene only on one of the two X chromosomes, because each female can have two chromosomes. So if she has the defective gene for the Duchenne muscle dystrophy, in order for her to manifest the disease, this gene needs to be present on both chromosomes, which is very unlikely, as we're going to see in a minute why it's so unlikely to have the defective gene on both X chromosomes. So females mainly will act as carriers. This means what? This means that a female will have only one copy of the defective gene on one X chromosome, and the other one won't have this defective gene. This is a normal X chromosome. It has no problem. Would she manifest the disease like this? No. Duchenne muscle dystrophy is not going to be manifested unless it's going to be present on both X chromosomes. All right? Why? Because simply on the other X chromosome, if I don't have the defective gene, I have a normal gene that will be covering the presence of the defective gene. But who will have a problem? Males. Males, again, they have an X chromosome and a Y chromosome. And remember, Y chromosome, it's a small chromosome. It doesn't carry much of the genes. So having an X chromosome that carries the defective gene responsible for the Duchenne muscle dystrophy, as a male, I don't have another normal gene to cover it, as I don't have another X chromosome to hide the presence of this defective gene. So if a female has the defective gene on one of her X chromosomes, we call this as a female carrier. She is a carrier of the disease. So she carries the disease, but the disease is not manifested on her. So the disease itself is not going to be affecting her. So what are my chances? If a normal male gets married to a carrier female, So a carrier female, either she's going to be producing a normal X chromosome or an X chromosome that carries the defective gene that codes for the Duchenne muscle dystrophy. And a normal male is going to be producing either an X chromosome or a Y chromosome in his sperms. So what are my chances here? I have XX, XY, XD, X, or XDY. So what are the chances on here of having a diseased child? Who will have the disease out of those four? The boy with the gene, exactly. So I'm representing 25% of the next population, 25%. So the mother did not show any symptoms of the disease. She carried the disease for her male children. Male children are the ones that will be manifesting the disease. Again, why this will not affect females? Because female gonna have two X chromosomes. If she has the defective gene on only one chromosome, it won't be manifested. It needs, for a female to show the disease, she needs to have the defective gene on both of the X chromosomes. So what is the problem here with the Duchenne muscle dystrophy? So we understand that females will carry the gene, but why is it very unlikely to have a female that will be showing the disease? So let's have a look on here at the timeline of the Duchenne muscle dystrophy. In Duchenne muscle dystrophy, you see on here, I have difficulty with my movements, weakening in the muscles, or weakening of the muscles is gonna be increasing over time. See this child is gonna have difficulty climbing the stairs. He's pulling himself up. See, those children who have the disease will start to develop spine deformity, as you see on here. So this child is developing lordosis, for example. Again, why? Because of the weakened skeletal muscles. So I can't keep up with my friends. I'm not able to contract the muscles as strong as the other kids. We'll have even difficulty standing up. We say here that the patient is as if he's climbing himself. Need to exert more effort to stand up. Here the kid is climbing himself, he's pushing himself up because of the weakened back muscles. Major spine deformities, as you see on here. So we're asking the kids here to move their arms up or to touch their nose. They will find difficulty performing those actions. With time, they will start to fail to be able to perform the basic muscle movements. So we're asking the kid here to move his arm up above his head or his hand above his head. Scoliosis is going to be present in almost 90% of the patients. You see on here is the lateral deviation of the spine. More difficulty in moving the arms. I'm not able to move my hand towards my mouth even. So those patients will need assistance in order for them to perform their basic daily activities. As the case gets worse, so I will need a ventilator for me because of the weakening of the respiratory muscles. I need also assistance in order for me to cough, to push any mucus that accumulates within the airways to prevent pulmonary infections. Most of the patients die within their 30s, early 30s or late 20s. Because of the weakening of the respiratory muscles. For Christina, yes, the enlarged cuff muscles, as you've seen for some of the kids in here. This is due to the accumulation of connective tissue. It's not muscle fibers that is forming those enlarged muscles. They will look enlarged, but they actually are so weak. All right, so I'm not sure if somebody is using a tablet, so you are marking on the screen on here. Can you please remove? I'm not sure where to remove it, where to remove this from. All right, I figured this out. All right. All right, so again, what we're looking at here is a homeostatic imbalance that affects the skeletal muscles. It's a sex-linked muscle disease. Usually females will be the carriers for the disease, and the disease affects their male children. Again, why it's very unlikely to see a female with the Duchenne muscle dystrophy, because males who carry the defective gene they have the disease. And most of those males will die at earlier age. If, let's say, I want to have a female with a disease, so in order for this female to have the disease, she needs to have 2 copies of the defective gene on both of her X chromosomes. So this won't happen unless a diseased male gets married to a carrier female for this X chromosome to meet this X chromosome, for the female to have 2 defective genes. And this is going to be very unlikely. So the diseased males won't live long enough to get married in most of the cases. All right, that's why it's so unlikely to have a female with Duchenne muscle dystrophy. Usually, patients of Duchenne muscle dystrophy will die of respiratory failure in their 20s. There is no cure for the disease. Viral gene therapy and infusion of stem cells to correct the dystrophin genes may show some promise in the future. But generally speaking, there is no cure for the disease. Moving on to another homeostatic imbalance, it's myasthenia gravis. Myasthenia gravis is an autoimmune disease. Auto, remember, means self. And autoimmune disease simply means that the immune system is going to be attacking the body self-antigens. So the immune system starts to target my own tissues. And in this specific disease, the immune system is going to be producing immunoglobulins that will be binding to the acetylcholine receptors. If you remember, this was the terminal end of the neuron. This is where you're going to be storing the neurotransmitters. And if you remember the neurotransmitter at the neuromuscular junction, this was my acetylcholine. Acetylcholine, in order for it to have an effect on the muscle cell, it did need to bind to its receptor in the sarcolemma. Here, the immune system is attacking those receptors by producing antibodies. So what's going to be the manifestation? If you're attacking the acetylcholine antibodies, the acetylcholine won't be capable to bind to its receptor. And remember, what was the effect of the acetylcholine when it binds to its receptor on the muscle? Any idea? What happens when you attach the acetylcholine to its receptor on the muscle cytoplasmic membrane? Open sodium channels stimulates the muscle cell, exactly. Remember, when the acetylcholine binds to its receptor, it opens sodium channels, allowing the sodium in, which will cause the stimulation of the muscle and the creation of an electrical impulse that travels along the plasma membrane. And this is gonna cause the opening of calcium channels, which will induce the muscle contraction. Remember, in order for you to contract the muscle, you need to release calcium from the sarcoplasmic reticulum into the cytoplasm. And this will allow the myosin subunits to bind to the actin and pull them to slide along the length of the thick filament. And this what results in the shortening of the length of the muscle cell. So without having acetylcholine binding to its receptor, this means you will not be creating this electrical impulse. You will not be enhancing the release of the calcium from the sarcoplasmic reticulum into the cytoplasm. So you will not be able to expose the binding sites on the actin subunits. You will not form the cross bridge between the myosin and the actin. You will not be sliding the filaments, and you will not be shortening the length of the muscle cell. And as a result, there will be no contraction. So what's going to be the manifestation here of the disease? If I failed to attach the acetylcholine to its receptor because the immune system started to produce immunoglobulins that will be targeting the acetylcholine receptors, there will be muscle weakening, drooping of the eyelids because I can't keep them open. I can't keep my eyes open all day long because in order for me to keep my eyes open, I need to send out signals all the time to my muscles, and the muscles need to respond by contracting. So in order for me to do this, I need to release a lot of acetylcholine to bind to the receptor. So if acetylcholine failed to bind to its receptor, you will not be able to conduct the signal to the muscle, so the muscles will fail to contract, and as a result, there will be drooping of the eyelids. As the disease gets worse, there will be difficulty in two of the most complicated tasks that we perform. Those are in swallowing and talking. So speech and swallowing, those are two of the most complicated tasks that we perform. They need lots of signals to be sent to the muscles, to not only one group of muscles, but to many groups of muscles, and they need to coordinate together in order to allow you to speak or allow you to swallow. So as we're going to see in this chapter and the next chapters, with the neurological homeostatic imbalances and the muscular homeostatic imbalances, two of the first functions that will be affected are going to be swallowing and talking. All right, because they need a lot of signals to be sent in a very brief time, a very brief period of time, and they need a lot of coordination to be taking place between this large group of muscles. Questions? Questions? All right, so starting today, our discussion of chapter 11. Chapter 11 covers the Chapter 11 covers the introduction of the nervous system. So first, what are the functions of your nervous system? Why do you have a nervous system? First function of the nervous system is to allow you to detect sensation. Sensory input. So now you can hear me. So the sensation here is going to be hearing. You can't see the board. So the sensations that you are detecting here is going to be vision, and so on. So nervous system will allow you to detect sensation. It allows me to have a sensory input. I'm collecting information from the outside through my receptors, and the nervous system will allow me to collect this information and will later process it. Second function of the nervous system is going to be the integration of those sensations. So, for example, I did mention something about the contraction of the muscle. This reminds you of something that you've studied yesterday, for example. So you did hear me. This is a sensory input. You're collecting the information from outside by hearing, and you are processing this information in your brain, and this is going to be considered an integration of the information. So you did hear something that I'm saying right now. This will be integrated with your memories. It's going to be integrated with something that you will recall later on, something that you've seen earlier if you were working in a hospital before or something like this. So integration of the information, and to give the information a meaning, is going to be taking place in my brain. Let's say you figured out that this information is important, so you decided to write this information down. So how would you write this information down? You're going to be conducting motor output signals to your skeletal muscles for them to contract and relax in very complicated patterns for you to be able to write down the information that you did hear right now and process for you to study it for the upcoming exam. So three main functions here for the nervous system. I'm able to detect changes in the outside environment. The change in the outside environment, this is what we call a stimulus. I'm able to detect that there is a change in the external environment, and what allows me to detect that there is a change are my receptors located in my skin if it was a touch sensation, in my ear if it was sound waves, if it was light, I will be detecting the light by receptors located within my eye, and so on. I'm able to collect this information. This ability is called sensation. This is my first function of the nervous system. Second function of the nervous system is going to be to process the information that you've collected, store the information, recall it whenever you need. So you see on here, this is going to be the second function of your nervous system, is going to be the integration. You decided to take notes. You're going to need to send out motor impulses that will be contracting your muscles in order for you to move your hand, finger, hold the pencil, and write down your notes. In this example on here, I've seen a glass of water. This is sensory input, visual input. I know that I want to drink. So I integrate what I see, which is a cup of water, with what I feel, for example. This is called the neural integration. I did integrate the information that I'm collecting from outside with something that I already have inside. And accordingly, I decided to grab this glass of water. So in order for me to grab the glass of water, I will be sending out motor impulses to my skeletal muscles that allows me to grab this glass of water. So again, three main functions of the nervous system. Those are sensory input. This is going to be the information gathered by your sensory receptors. It might be something internal or external. Integration. This is the interpretation of the sensory input. The sensory input has a meaning for you. So if this is the first time that you see a glass of water, would you know that this is a glass of water that you can drink? What do you think? This is the first time in your life to see a glass of water. Would you know that this is something that you can drink? What do you think? No. So did I see this glass of water? Yes, you can see it. No problem. You have sensory input. But as you don't know that this is a glass of water, there will be no matching of the input information with what you already know. There is no interpretation of what you see. So for example, if I'm writing down something like this, can you see what I'm writing down on here? Can you see this? Hieroglyphics. Right? So can you see? Yes, no, yes. Do you understand what this means? This is the difference between sensation and integration. Sensation is your ability to detect that there is a change. What is the change? It was all black like this, and you did detect that I'm drawing figures, but you cannot interpret those figures. There is no integration with the sensor between the sensory input and the previous information that you've learned. If you've seen hieroglyphics before, you would be able to match those figures. Yeah, they are hieroglyphics. So you would be able to match what you see with what you know. There is some degree of integration. But can I read this? No. Can I understand what this means? No. But can I see it? I can see it. I have no problem seeing those figures. I have sensation. But can I interpret what I see? Can I give a meaning to what I see? What do you think? Can you give a meaning to what you see on here? What did I write down? What this means? Bird eating. Bird lives in the tree by the water. Yeah, you're close. You're close. So actually, this is my name here. M-I-N-A, Mina. All right, so here's this an owl, leaf, water, and a bird. A bird is A, water is an N, leaf is an I, and the owl is an M. All right, so now when you see an owl, you will know that this M. When you see a leaf like this, it's going to be an I. Water, N. Bird, A, and so on. So now you can match those shapes with something that you already know. Does this make sense now? Does this make sense? All right, so sensation again is your ability to know that there is a change, to detect the stimulus. Light, sounds, change in your blood pressure, change in your blood sugar level, all those are changes, all those are stimuli. They are detected, and the ability of detecting the stimulation is what we call a sensation. This is the sensory input. My ability to interpret it, to give it a meaning. So if I have a low blood pressure, let's say I have receptors here in my blood vessels that are detecting the degree of stretch applied to the wall of the vessel. If I do have low blood pressure, what's going to happen to those receptors? They will detect that you do have a reduction in the degree of stretch of the vessel. This is the information that you're going to be sending to the brain. This is your sensation. Your brain interpreted as low blood pressure. It translates the signals that it did receive, that there is a reduction in the degree of stretch in the wall of the artery, as a reduction in the blood pressure. And accordingly, it's going to be responding, sending out a motor output. So in this case, it's going to be going not to my skeletal muscles, it's going to be going to my smooth muscles. For the smooth muscles to contract, constricting the vessel. And as I constrict the vessel, this increases my blood pressure to normal. So you see on here, my ability to detect that there is a change. I didn't drink any water today. I have low fluid intake. So my blood pressure is supposed to drop. But what happens? Once I have a slight decrease in the blood pressure, there will be a stimulation of those receptors, and they will send the sensory input to your brain. And your brain will do what? It integrates the signals it receives with what it's supposed to respond with. The integration here, it gives it a meaning. The meaning of having reduction in the degree of stretch in the wall of the artery means for the brain that you have or about to have a drop in your blood pressure. So it decides, now I need to respond. How would I respond to a drop in the blood pressure? Sending out motor impulses to whom? To the smooth muscles of the blood vessel for them to contract, constricting the blood vessels, and this will be increasing the blood pressure to normal. Easy, easy. All right, great. So looking here again, third function of the nervous system is the motor output, which is going to be the activation of the effector organs. Those effector organs might be either muscles or glands. And their response will be either to inhibit the original stimulus or to enhance the original stimulus. If you remember, this is the feedback that you get. We did study back in chapter 1, a positive feedback and the negative feedback. All right, so again, three main functions on here for the nervous system: sensory input, my ability to detect that there is a change either in the external or internal environment. This is going to be detected and conducted to my central nervous system. This is where you're going to have the integration. The interpretation is going to be taking place in your central nervous system. Central nervous system is going to be responding according to the signals that it receives by sending out motor impulses to the effector organs. Again, effector organs are either going to be muscles or glands. Muscles, either skeletal muscles, smooth muscles, or cardiac muscles. Those are the main effector organs that I will be acting on. Any questions? Any questions? Anatomically, we do classify the nervous system into two main parts. We have the central nervous system, which is formed by the brain and the spinal cord. And the peripheral nervous system, which is going to be formed by the nerves going in and out of the central nervous system, either from the brain. So we call the nerves on here, those are my cranial nerves, or they are conducting signals in and out from my spinal cord. So we're going to call those are my spinal nerves. So we classify the nervous system anatomically into two major parts. Central nervous system, again, this includes the brain and the spinal cord. This is my CNS, central nervous system. Peripheral nervous system, or PNS, is going to be formed by the nerves going in and out of the central nervous system. If I am conducting signals in and out directly from the brain, those are called my cranial nerves. If I'm conducting signals in and out directly from the spinal cord, those are my spinal nerves, and both together are considered part of my peripheral nervous system. the peripheral nervous system is going to be subclassified into two major functional divisions. So those nerves that are conducting signals in and out of my brain and spinal cord, forming my peripheral nervous system, those might either be conducting motor signals or sensory signals. So we classify the nervous system, the peripheral nervous system, those nerves into either sensory nerves or motor nerves. This is a functional classification. So some of those nerve fibers are conducting sensations, sensory, or they are conducting motor impulses, they are motor nerves. So sensory nervous system is going to be called another name, which is the afferent. Afferent, I'm conducting the signals to the brain, to the control center. If I'm getting sensations from skin, skeletal muscles, joints, I will classify this part of the sensory nervous system as somatic sensory nervous system. So sensory nervous system might be somatic if you are getting signals from skin, skeletal muscles, or joints. It might be visceral afferent, visceral sensory, if I'm collecting the signals from my visceral organs, from the stomach, for example, from the gastrointestinal tract, from the blood vessels, all the nerve that are collecting information from the visceral organs are going to be visceral, part of the visceral sensory nervous system. Any problems so far? All right. Moving on to what if I'm conducting signals from the brain to the effector organ, to the muscles or glands. The nerves that would be responsible to conduct the signals from the central nervous system to the effector organs, to the muscles and glands, those are going to be forming my motor nervous system. And the motor nervous system is going to be subclassified into somatic or autonomic nervous system. Somatic is going to be sending out motor signals to your skeletal muscles. If you are sending out motor signals to your smooth muscles located in the wall of hollow organs, cardiac muscles in the wall of the heart, or to the glands, all those are gonna be under the autonomic nervous system control. So think of it in a different way on here. So which one do you have a voluntary control on? Your skeletal muscles or your smooth muscles, cardiac muscles, and glands? Can you decide right now to, for example, increase the sweat secretion or reduce the heart rate or increase the force of contraction of your heart or reduce the motility of your gastrointestinal tract or to dilate your pupil or relax your blood vessels or secrete insulin from the pancreas? Can you decide to do any of this? All of those are under involuntary control. So what is the part of the peripheral nervous system that's going to be responsible to send out the signals to those effector organs that you don't have any control on, smooth muscles, cardiac muscles, and glands? What do we call this part of the nervous system again? It's anybody? What do we call the part of the nervous system that you don't have any control on, any voluntary control on? Again, it's my autonomic nervous system. Autonomic nervous system. This classification needs to be crystal clear in your mind because those are the upcoming chapters. So in this chapter, we have a general introduction of the nervous system. In the next chapter, we are discussing the central nervous system. Next chapter is going to be the peripheral nervous system. The following chapter is going to be the autonomic nervous system. So this classification needs to be crystal clear in your mind. Because if you don't understand the difference between autonomic nervous system, somatic motor nervous system, somatic sensory, somatic visceral, you're going to be in trouble until the end of the semester. So again, if I'm collecting sensory input, we classify the nerves as sensory nervous system. All of them together are considered together as sensory nervous system. If I'm collecting the signals from skin, skeletal muscles, and joints, this is going to be somatic sensory nervous system. If I'm collecting signals from the visceral organs, this is going to be visceral sensory nervous system. What if I'm sending out motor signals to the effector organs? Again, what effector organs would you have responding to those motor signals that you get out from your central nervous system? Either you're affecting muscles or glands. If I'm sending out the signals to skeletal muscles, as I have control on the skeletal muscles, they are under voluntary control. So the part of the nervous system here is what we're going to classify as somatic motor nervous system. For all the rest of the muscles and glands, you don't have any control on them, so they are under involuntary control. We classify the part of the peripheral nervous system conducting the motor signals to your smooth muscles, cardiac muscles, and glands as the autonomic nervous system. Any problems? Any problems? Is this clear? right? Very, very important to have this all the time in the back of your mind. Very, very important. When we're going to be discussing the autonomic nervous system in chapter 14, we're going to learn that we do subclassify the autonomic nervous system according to the response into either parasympathetic nervous system or sympathetic nervous system. So again, if we're looking here at the classification of the different parts of the nervous system, anatomically, we divide the nervous system into central nervous system that includes the brain and spinal cord, and peripheral nervous systems. This includes the nerves carrying either signals to or from my central nervous system. If I'm carrying sensory signals from the organs to my central nervous system, we classify this as a sensory division. We subclassify the sensory division of the nervous system according to where you're getting the sensations from. Are you getting the sensations from skin, skeletal muscles, or joints? This is going to be somatic sensory nerve fiber. If you're conducting sensory impulses from visceral organs, the fiber here is going to be a visceral sensory nerve fiber. What if you are sending out signals? If I'm sending out the signals from the nervous system to the effector organs, this is going to be my motor division of the peripheral nervous system. And according to whom you're sending the signals to, if you're sending the signals to the skeletal muscles, this is going to be somatic motor. If you're sending the signals to cardiac muscles, smooth muscles, or glands, this is going to be the autonomic nervous system division. And as you don't have control on the autonomic nervous system, you're going to have two subdivisions of the autonomic nervous system to regulate the responses of those organs. So for example, one is going to be increasing your heart rate. So what's going to be the function of the other subdivision of the autonomic nervous system, in your opinion, to have the most accurate responses? If one is increasing my heart rate, what is going to be the function of the other one? Will the other one also increase the heart rate? Do you get my question? So the other division should be reducing the heart rate for me to have the best regulation of the heart rate. So as we're going to learn, sympathetic nervous system is going to be increasing the heart rate. Parasympathetic nervous system is going to be decreasing the heart rate. One is going to be enhancing the urinary bladder to contract, releasing the urine to the outside. And the other one is going to be inhibiting the urinary bladder from contracting. So in order for me to have the best regulation of those organs, heart, smooth muscles, and glands, I have again two subdivisions of the autonomic nervous system. Sympathetic nervous system and the parasympathetic nervous system. Questions? Questions? All right, so first question is the in-class activities for today. It's going to be here. So while you're studying for an exam, you reach for a beverage to extend your arm. What part of the nervous system is going to be activated? So this is first question today for the in-class activities. What do you think? Somatic. Why? Because when you decide to extend your arm, you're moving what kind of muscles? You're moving skeletal muscles. And again, what part of the nervous system going to be sending out signals to my skeletal muscles? This is going to be my somatic nervous system. Is this clear? Clear? All right, so let's see another one. Again, this is going to be another in-class activities. This is our second question for today. The door slams shut loudly and you flinch. After a few seconds, you realize that your heart is beating very rapidly and forcefully. So your heart is beating faster. This response is a result of your blank nervous system. My heart is contracting faster. I have an increase in my heart rate. So what part of the nervous system is going to be responsible to send out signals to your heart? See 3 responses out of 21 people attending. 5 responses. Am I sensory or motor first? Is the heart a muscle or a receptor? It's a muscle. So if I am sending the signal to the muscle, is it going to be a motor or sensory? An afferent or an efferent? You're sending out a signal. I'm sending out. So you see on here, your heart is beating very rapidly. So beating very rapidly, this means it's contracting. It's contracting. And this is a response. This response is resulting, is a result of your what part of the nervous system? Is it going to be sensory? It's not sensory input. It's not sensory input. I'm telling you that your heart is contracting, not you can see, not you can hear, not you feel. So this is not a sensory input. I'm not collecting information. It's a response. So I am performing a muscle contraction. So muscle contraction or a glandular secretion, it's a result of the activation of the motor division of the peripheral nervous system. So motor, and we did subclassify the motor nervous system into what again? Into either somatic or autonomic. Am I sending the signals to the skeletal muscles or one of the three on here? Smooth muscles, cardiac muscles, or glands? Which one am I sending the signals to? Skeletal muscles, smooth muscles, cardiac muscles, or glands? Cardiac muscles. So what is the subdivision of the nervous system which will be sending out the signals to your cardiac muscles? This is going to be your autonomic. All right. Is this clear? Is this clear? All right. So let's have a break for 10. We come back, discuss more about the nervous system. We can have a break for 10. We come back to discuss more about the nervous system. Right. Any questions before we go to the break? Any question? All right, great. So let's have a break for 10 and we'll come back to complete our discussion for today. All right, welcome back. For Harris, the lab exam, as we've mentioned earlier, it's going to be available for you to complete for 1 week starting today until Tuesday. So it's going to be available for 1 week. You can go to the homepage on the Canvas course and you will be able to find it under the updated lab exams. It has been posted today. It will be available for you to complete for 1 week. You have one attempt, you have 50 questions. Each one is going to be out of 2 points. This gives you a total of 100 points, and you're going to have 75 minutes as a time limit for the test. It's going to be in the form of multiple choice questions. So you're going to have a list of choices for each of the markings. 75 minutes, I believe, in my opinion, is more than enough. I've completed it in around 12 minutes. So you should be able to, everybody should be able to answer the test in not more than 75 minutes. Does this answer your question? Has anybody else has a question concerning the exam? So please, if you know somebody who's not attending neither the live or the recorded lecture, please let them know, because I won't figure out who's going to be missing the live exam and who's going to be waiting until the last minute to get it completed, until they actually miss the due date. All right, so if you know somebody who's not making it to the live, or he's not making it to the live and not attending the recorded session, please let them know this information. I've posted this information earlier today as an announcement on Canvas, but it seems for me that some people are not checking their Canvas. All right, so please, if you know somebody who's not attending those live meetings, or not attending, or not watching the recorded sessions, please let them know of the due date of the muscles lab exam. All right, so looking here at the histology of the nervous tissue. If you remember back when we studied the tissues, we've seen 2 types of cells forming the nervous tissue. We've seen cells with a lot of dendrites, filaments coming out of the cells like this. We call those are my nerve cells or my neurons. And in the background, if you remember, we did see nuclei within cells in the background, and we did call them back in chapter 4, those are the supporting cells. Not sure if you remember this. You remember then how the nervous tissue did look like under the microscope? We've seen neurons, which are the cells with processes coming out, and we've seen in the background many cells, which are the supporting cells. And if you remember back in chapter 4, I told you that we're going to discuss those supporting cells in details when we get to the nervous system. And here we go. Those supporting cells are called the neuroglial cells, or the glial cells. We have different types of glial cells or supporting cells that we're going to be studying in this chapter. First one is called the astrocytes. Next is microglial cells. ependymal cells, oligodendrocytes, satellite cells, and the Schwann cells. Those are the neuroglia or the glial cells that did appear in the background of the neurons, of the nerve cells. Let's start our discussion with the supporting cells. If you're looking on here, you can see a cell with many arms. This cell looks like a star. You see on here. So a cell that looks like a star, we call it astrocyte. Astro is star. Cyte is cell. So the cell that looks like a star, this is my astrocyte. And if you see on here, astrocytes are connecting what to what? They are connecting my blood vessels to the nerve cells, to the neurons. So what is the function here of the astrocyte? So let me ask you another question for the function of the astrocyte to make sense. So if you are the CEO of a big company, CEO of an energy company or an electronics company, you are performing very complicated tasks. You are following the stock market, you are following the engineering department to enhance the technology that you're using for your products and so on. The marketing department, you're following up with them to check how many did you make this quarter and so on. So you are highly specific to what you're doing. You are so busy in what you're doing, same like the neuron, same like the nerve cell. Would you have time to cook for yourself? What do you think? You are CEO of a big company, Apple, let's say. Do you think you're going to have time to cook for yourself? Or rather than cooking, you're going to be doing more money. You're going to be doing more money, definitely, right? So I don't have enough time to cook for myself, so I need to hire somebody to cook for me. So the same concept here will be applied to the nerve cells. Nerve cells are highly specialized excitable cells. They are performing complicated tasks that no other cell in your body can perform. So they are hiring the astrocytes to cook for them. The astrocytes going to be responsible to get the nutrients, to perform the energy metabolism, and to transport those products to the nerve cells. Because as a nerve cell, I don't have much time to cook for myself. Another function for the astrocyte is to prevent any toxins from moving from your blood to your nerve cells. So I am part of what we call the blood-brain barrier, or BBB. So as a CEO of a big company, you might hire some bodyguards. So who's going to be cooking and serving as a bodyguard here for the nerve cells? Those are my astrocytes. You don't want to have a toxin passing from your blood to your nerve cells. Why? Because those nerve cells are permanent cells. You can't replace them. So if a person lost a nerve cell, it got damaged, it can't be replaced. It can't perform apoptotic division and can't get replaced. So again, what's going to be the function here of the astrocyte? Astrocyte will be regulating the energy metabolism and will be transporting the blood-borne material to the neurons, acting as a mediator between the blood vessels and the nerve cells. They are part of my blood-brain barrier, not allowing everything to pass from the blood to the nerve cells. Make sense? Make sense? Anybody has a problem with astrocytes? All right, great. Moving on. Moving on to another neuroglial cell. This is my microglial cell. And as you see on here, the microglial cells are micro. They are smaller in size compared to the nerve cell. That's why we call them microglial cells. They are defensive cells located in the CNS, and the main function on here is to attack any pathogen that find finds its way to the neurons. So this is another bodyguard, but this time this is a bodyguard specialized to attack any pathogens, like the tissue macrophages. Another type of neuroglial cells, those are my ependymal cells. And as we're going to be discussing later today or at the beginning of next class, we're going to learn that inside your brain, you've got ventricles. And those ventricles that will look like this are going to be allowing the circulation of a fluid inside, and also this fluid is going to be circulating around the brain and the spinal cord. So again, inside the brain in cavities called the ventricles, and inside the spinal cord in a space called the central cavity. Also around this brain and spinal cord, you've got fluid, and this fluid is what we call the cerebrospinal fluid, or CSF. Cerebrospinal fluid is the fluid circulating within my ventricles and in the central canal of the spinal cord, as well as around the brain and the spinal cord. So if you look at the heart, at the blood, what allows the blood to move in your vessels? It moves because you've got cardiac contraction, right? So the cardiac contraction will be pumping the blood into your vessels. This is what allows the circulation. But do I have a heart in here to allow the circulation of the CSF? Inside the ventricles, around the brain? No. So the CSF actually gonna be circulating by the propulsion effect of the cilia of the cells lining those ventricles. So if I get closer, lining those ventricles, you're gonna have cells with cilia. And those cilia will be pushing the fluid for it to circulate inside the ventricles and around the brain. And we call those cells with cilia lining the ventricles, those are my ependymal cells. So again, how many glial cells we've seen so far? We've seen 3 of them. One that looks like a star, we call this is my astrocyte. It's going to be helping with the energy metabolism for the neurons, also acts as a barrier between your blood and your brain cells. Another glial cell that we've seen, this is my microglial cell. It's a defensive cell that will be attacking pathogens. A third glial cell, a third supporting cell located in the nervous system, this is going to be my ependymal cell. And ependymal cells, those are cells, as you see on here, with cilia. And the ciliary propulsion is what's going to be pushing the fluid inside the ventricles of the brain and around the brain. And again, this fluid is what we call the cerebral spinal fluid, or CSF. Another neuroglial cells that we can see in the central nervous system is going to be my oligodendrocyte. Oligo means I have few. Dendrocyte, dendro means processes. So this is a cell on here that has few processes coming out. That's why we call this is my oligodendrocytes. Cells with few processes, oligodendrocytes. They are located in the CNS, and their main function is to surround the nerve fibers with myelin sheath. What is the myelin sheath? So if I have a charging cable for my phone on here, when I hold this charging cable in my hand, do I have any electrical transmission to my hand? What do you think? Is there any electrical transmission? If this cord, this charging cord of my phone is connected to the electricity on one side, and it's connecting to my phone on the other side, holding the cable will cause any electrical transmission to my hand. What do you think? No. Why? Why there is no transmission of the electricity to my hand? Because you are not holding the part that conducts the electricity. The part that conducts the electricity is located on the inside of the cord, the metal part. What you're holding is the plastic wrapping of the charging cord. That will be acting exactly as an insulator. So what will be preventing the nerve fibers on here from conducting their electrical impulses to nearby tissues? Simply they are insulated. And again, what's going to be insulating those nerve fibers? What's going to be covering those nerve fibers, preventing the conduction of the electrical impulses to the surrounding? It's going to be the myelin sheath. So what acts as here the plastic part or the rubber part around the copper in your charging cord? What acts as the plastic wrapping of the charging cord is going to be the myelin sheath. All right. What is the name of the cell again that's going to be responsible for the formation of the myelin sheath that's going to be surrounding the nerve fibers in the CNS, in the central nervous system? Those cells are called my oligodendrocytes. Any difficulty? Any difficulty? Another type of neuroglial cells, those are going to be the Schwann cells. Schwann cells will be acting the same way like my oligodendrocytes. They are going to be responsible for the formation of the myelin sheath wrappings of the nerve fibers. So I'm forming the myelin sheath around the nerve fibers, but they are going to be located in the peripheral nervous system. So again, what is the cell responsible for the formation of the myelin sheath in your central nervous system, in your brain and spinal cord? This is going to be the oligodendrocytes. What is the cell that's going to be responsible for the formation of the myelin sheath around the nerve fibers in your peripheral nervous system? Those are my Schwann cells, right? Satellite cells to a less extent. Schwann cells are the ones here building up the myelin sheath. So again, what do we call the ones in the CNS forming the myelin sheath? Oligodendrocytes. What do we call the ones that will be in the peripheral nervous system building up the myelin sheath? Those are my, again, Schwann cells. Easy. All right, so looking at the neurons, the nerve cells. Nerve cells are long-lived cells. They live 100 years or more. They are amitotic. What I mean by amitotic, they do not perform a nuclear division, and thus they don't perform a cell division. So there is no nerve cells that can divide to form two identical copies of its own. They have a very high metabolic rate, and this explains why it needs continuous supply of oxygen and glucose. So if a person is shocking, so I did allow something to pass into my airways, blocking my airways. So what would I develop? I will first thing that's going to happen to me, if it was not resolved, I will lose consciousness. If I am a diabetic patient and I take an insulin shot early in the morning and I didn't have breakfast, so I received a phone call, I forget about eating, so I didn't eat anything and I got an insulin shot. So what's going to happen to my blood glucose levels? In this case, my blood glucose will drop. And this might result in a hypoglycemic coma. I will lose consciousness because of the drop in the blood glucose levels. All right. So again, why your brain can't function in case of low oxygen or in case of low glucose, because it has a very high metabolic rate. It needs a lot of energy to function. All the time I need ATP. All the time I need ATP. And the best way to get ATP is by breaking down the glucose in the presence of oxygen, if you remember. What do we call the process by which we're going to be breaking down the glucose in the presence of oxygen? You remember from our last meeting? Yeah, more specific than glycolysis. Aerobic glycolysis. Aerobic glycolysis, exactly. Because glycolysis might be in the presence of oxygen and in the absence of oxygen. We call the one that takes place in the absence of oxygen. This is my anaerobic glycolysis. And for the ones that takes place in the presence of oxygen, this is the aerobic glycolysis. If you remember, the ones that's going to be taking place in the presence of oxygen yields much more ATP, 32 to 36 ATPs, compared to the anaerobic glycolysis, it yields only 2 ATPs. Easy. Questions? Any questions? All right, so looking here at the structure of my nerve cell, if you see the nerve cell has a cell body, this is where you're going to keep the nucleus of the cell. This is my cell body. And the cell body, coming from the cell body, we've got short tapering processes. We call those processes are my dendrites. And coming also from the cell body of the neuron, we've got a long process. We call this the axon. At the junction between the cell body and the axon, we've got a bulging part in here in the cell body. This is where the axon gonna be formed and attached. This is called my axon hillock. The axon of the nerve cell will have terminal processes like this, and those terminal processes are what we call the telodendria. Telo is tail, and dendro are processes. So the tail processes, which are the processes coming from the end, coming from the terminal end of my axon, we call those are my telodendria, or the terminal end. Each of those telodendria will have those boutons. We call them either the synaptic nodes or synaptic boutons. Those are those bulging ends located at the end of my telodendria. Each axon, if you remember, it's gonna be surrounded by an insulating material. We call this is my myelin sheath. And the cells responsible for the formation of the myelin sheath surrounding the axons in the peripheral nervous system, those are my Schwann cells. So here, those are the Schwann cells surrounding the axon of my nerve cells by the myelin sheath. If you notice on here, unlike the charging cord that we've seen earlier, charging cord, the copper part that will be conducting the electricity is fully covered with the plastic. All of it is covered from outside by this plastic. But if we're looking at the axon of the nerve cells, you will see, do I have a continuous covering of the axon of the nerve cells? What do you think? Do I have a continuous covering of the axons of the nerve cells? No. If you notice on here, I have parts that are not covered with myelin sheath or Schwann cells. We call those interruptions in between the Schwann cells and the myelin sheath. Those are the nodes of Ranvier. So again, what we're looking at in here is the structure of the nerve cells or the neuron. The neuron will have a cell body. Coming out from the cell body, we've got those short tapering processes. Those are my dendrites. Coming from the cell body, also a long process, we call this is my axon. This bulging segment where the axon gets its origin from, we call this is the axon hillock. The terminal processes of the axon in here, those are my telodendria. And if you notice, there will be those bulging parts in each of those telodendria. We call those are my terminal or synaptic knobs or boutons. Also, I want you to notice that the axon is covered with myelin sheath, which gets formed by the Schwann cells. And the myelin sheath is not fully covering the axon. We've got interruptions in the myelin sheath. And again, the interruptions here of the myelin sheath is what we call the nodes of Winfield. All right, so for another in-class activity, please name the different parts of the nerve cell. We've got 2 questions before. Here is going to be another question as part of your in-class activities. All right, so this is going to be number 3 because we've got the first 2 questions already. So this is going to be number 3. Four, 5. Here's the whole thing is called what? This is going to be number 6. Those terminal processes are going to be number 7. The secretory parts of those terminal processes are going to be 8. The covering and the cell forming the covering of the axon is going to be number 9. And the interruptions here of the axon coverings, those are called what? This is going to be number 10. And of course, this is going to be the nucleus here. Again, this is going to be questions number 3 to number 8 to number 10 for the in-class activity for today. Anybody needs more time here before we move on? All right, so moving on to the function of the axon. Axons are going to be the conducting region of the neuron. This is where you're going to be conducting the signal. from one nerve cell body, the cell body of the nerve cell to the next nerve cell or the effector organ. They will be responsible for the generation and the transmission of the electrical impulses, and those electrical impulses might be traveling from the cell body of the nerve cell towards the telodendria. We call this as an anterograde electrical conduction. And I can conduct the electrical impulse from the telodendria backwards towards the cell body, and we call this as a retrograde electrical conduction. So again, we can conduct the signal, the electrical signal from the cell body to the telodendria. This is an anterograde conduction. We can conduct the signal from the telodendria to the cell body of the nerve cells. This is a retrograde conduction. Myelin sheath is going to be responsible to protect and electrically insulate the axons, and they will be responsible to increase the speed of the electrical transmission, as we're going to see in a second how. How do you form the myelin sheath? The myelin sheath is simply a lipid covering the axon. This lipid is simply the plasma membranes of the Schwann cells. So in order for you to cover the axon, one Schwann cell is going to be rotating around the axon, wrapping the plasma membrane, and will squeeze out its cytoplasm. So the remaining part of the cell is going to be simply the plasma membrane. So let's say on here, if this is my Schwann cell, it's going to be surrounding the axon like this, and starts to squeeze the cytoplasm to the outside. So what's going to be remaining outside around the axon are going to be the plasma membranes of my nerve cell. And all this fluid that got squeezed outside of the cell, this is what we're going to call the neurilem. All right, so again, the myelin sheath is simply the plasma membranes of my Schwann cells. What are the nodes of Ranvier? The nodes of Ranvier are the gaps located between the adjacent Schwann cells. So if you see on here, as we've mentioned earlier, this interruption in the continuity of the myelin sheath, those gaps between the adjacent Schwann cells are what we call the nodes of Ranvier. Do we have any nerve fiber axons that are not surrounded by myelin sheath? Yes, we have unmyelinated nerve fibers or axons. They are thin nerve fibers. They are not surrounded by myelin sheath. And this is due to the scarcity of the Schwann cells. So I have one Schwann cell that's going to be surrounding 15 unmyelinated axons. So I don't have enough Schwann cells that are going to be surrounding my axons. All right, we're going to be skipping the classification of the nerve cells for now to move on to one of the most important topics in this semester. This is going to be your essay question for the nervous system lab exam. All right, so how do I conduct an electrical signal? How do I form an electrical signal? What is the form of electrical signals that I will be conducting in my nerve cells? An electrical signal is simply a change in the membrane polarity. If you remember how your computer devices are communicating with one another, they are sending codes in the form of zeros and ones. 0 means I'm not conducting an electrical signal. 1 means I'm conducting an electrical signal. So what is the 1? What is the 0 for me as a nerve cell? Nerve cells, like any other cell in your body, they have much more potassium on the inside, much more sodium outside of the cell. But inside of the cell is going to be more negatively charged because I have much more anions. I have inside the cells more negatively charged ions. So in resting state, if the nerve cell is not conducting an electrical impulse, the outside of the cell membrane is going to be more positively charged compared to the inside of the cell membrane is going to be more negatively charged. So in a resting state again, in a resting state, the nerve cell is going to be more positively charged on the outside of the membrane, and it's going to be more negatively charged on the inside of the plasma membrane. What is the ion distribution again? Outside of the cell, you're going to have more sodium. This is the most common cation located outside of the cell. And in the intracellular fluid in the cytoplasm, the most prominent cation is going to be my potassium. If I measured the difference between the charges on the inside and the outside of the plasma membrane of a nerve cell in a resting state, I will see that the inside of the cell membrane is more negatively charged than the outside by a value of 70 millivolts. So if I measure the difference between the inside and the outside, I will see that the inside of the plasma membrane is going to be more negatively charged than the outside by 70 millivolts. If I've got a stimulus, let's say this is the nerve cell, this is a nerve fiber that will be excited to detect touch sensation. So if I did touch a surface with my skin, what's going to happen to the nerve cell? As you are applying a stimulus to the nerve cells will need to respond if they did get enough stimulation. Do you remember what do we call the minimal stimulus to which you're going to be responding? Threshold stimulus. So if, for example, a mosquito is moving on your skin, would you feel the touch sensation? Would you feel anything moving on your skin? In most of the cases, no. In most of the cases, no. Why? Because simply this doesn't create enough stimulation of the receptors responsible to detect touch. I didn't create a threshold stimulus. I don't have a threshold stimulus. It is not threshold yet. So in response to any stimulation, how your nerve cells gonna be responding? They will start to open sodium channels. I open sodium channels. So what's gonna happen? Where do I have more sodium, outside or inside of the cell? Can you see on here? Where do you have more sodium? Outside of the cell. So if I get enough stimulation or any stimulus, gonna open some of the sodium channels. So what's gonna happen to the sodium? Like anything, remember, anything gonna be traveling from high concentration gradient to low concentration gradient. So where do I have a high concentration of the sodium again? Outside of the cell. Compared to inside of the cell, I have very small amounts of sodium. So what's gonna happen? The sodium gonna be moving from outside to the inside of the cell. And is this a cation or an anion? Is this positively charged or negatively charged ion? What do you think? Sodium. It's a cation. So it's gonna be doing what to the inside of the cell membrane? Inside of the cell membrane in resting state, remember, it was more negatively charged. So now when I allow the sodium in, now the inside of the cell membrane will become more positively charged. This is a change in the membrane polarity. What I mean by polarity, polarity means I have two poles. This is my cell membrane. Cell membrane has two sides. It has two poles. One is more negative, one is more positive. This is the polarity. This is my resting membrane polarity. And again, resting membrane polarity is inside is more negatively charged than outside by a value of 70 millivolts. Inside is more negative compared to the outside by 70 millivolts. This doesn't mean that you only have negative charges inside and you only have positive charges outside. This means that the inside is more negative than the outside by 70 millivolts. All right, so inside you have cation, outside you have cations. But inside has much more anions, more negatively charged ions compared to the outside. That's why inside in a resting state, it's more negatively charged. So again, what is the value of the resting membrane polarity? If I measure the difference between inside and outside of the cell membrane during rest, what is the RMP here? Resting membrane polarity equals to negative 70 millivolts. Exactly. This is a zero in the code that you're trying to send. In order for me to send the signal, I need to get enough stimulation. a threshold stimulus. If I get a threshold stimulus, what's going to happen? I will open enough sodium channels that will allow the sodium influx. The sodium is going to be moving from outside of the cell to the inside of the cell. And this will be responsible of losing the resting polarity. I have lost the membrane polarity. This is what we call depolarization. Like composition, what is the opposite of composition? What's the opposite of composition? Decomposition. So I started by polarity. I have lost my polarity. Inside is negatively charged. I lost the negative charge on the inside. Inside is becoming more positively charged. So I've lost my polarity. What we call the phase on here in which you're going to be losing the membrane polarity is depolarization. Depolarization. I have lost my membrane polarity. It's a depolarized state. Inside is becoming again in a depolarized state, more positively charged or more negatively charged. What do you think? Inside of the cell membrane is becoming more positively charged or more negatively charged? More positively charged, more positively charged. And what is the ion responsible for the depolarization of the cell? Sodium. Sodium. So I started by having a 0. This is my resting membrane polarity. And now I've got my 1 in here. I've got a signal that is conducted along the cell membrane. This is my depolarization. In order for me to send another signal through the same cell, so I need to do what? Can I become positively charged on the inside compared to the outside while I am already positively charged? What do you think? So can you open a door and this door is already opened? Can you open an open door? Or can you close a closed door? No, you can't. So in order for me to create another signal, what I need to do, I need to go back to my initial membrane polarity, to my resting membrane polarity. I need to go back to the negative 70 millivolts. But in order for me to do so, I need to do what? I need to lose positively charged ions from inside of the cell to the outside. I need to reverse my membrane polarity. I need to get back my membrane polarity that I've lost, allowing the sodium in. This is how a sodium channel looks like. It has 2 doors like this to allow the sodium in. You're gonna open the outside door that is closed. in order to stop the influx of the sodium, in order to not get any more sodium in, I will close the internal gate. So this is a channel that has two gates. Outside gate opens to allow the sodium in. Inside gate will close to prevent any further influx of the sodium. So in order for me to return back to my polarized state, to return back to my resting membrane polarity, this is my resting membrane polarity. Inside should be negatively charged another time compared to the outside. And I did allow some of the sodium into the cell, but still I have most of the sodium located outside in the extracellular fluid. Most of the sodium is still located outside. Some of it that did pass when I opened those channels did pass to the inside of my cell. So if you want to go back to the resting membrane polarity, to the polarized state inside to become more positive than the outside, I need to get back to the negative 70 millivolts. So you do have two options. Either you move the sodium out, or you move this potassium out. Why do you need to move a positively charged ion from inside of the cell to the outside of the cell? Because you want to reduce the number of positively charged ions on the inside for you to get back your resting membrane polarity, for the inside to return back to its negative state. So what positive charges do I have inside? I have potassium inside. I have small amounts of sodium. Which one gonna be easier for you to move from inside of the cell to the outside of the cell? What do you think? Why potassium? Why you don't get this sodium back to where it came from? In order for you to depolarize, you allow the sodium in. So why I don't move it back out? Because simply it's gonna be trying to move against the concentration gradient. You're gonna be trying to move the sodium here from its low concentration inside the cell to its high concentration outside of the cell. And this is gonna need ATP, it's gonna require energy, and more importantly, it's gonna take longer time. So which one gonna be much faster to regain your resting membrane polarity is for you to open potassium channels rather than sodium. You close the sodium to not get any further influx of the sodium in the cell. In the same time, you're gonna need to open the potassium channels for the potassium to travel from inside of the cell to the outside of the cell. And when I lose those positively charged ions from inside of the cell, the inside of the cell will become more negatively charged another time. I did regain my polarized state. So we're gonna call this is my repolarization. So how do you conduct an electrical signal through the nerve fiber? You start first by a cell that is not conducting a signal. I don't have a signal. Inside is more negatively charged than the outside of the cell membrane. We call this state is my resting membrane polarity. Once I get enough stimulation, I open enough sodium channels. And once I open those sodium channels, this will cause a cascade of opening of the sodium channels throughout the length of the nerve fiber. This allows the nerve impulse, the electrical impulse, to propagate. What is the form of electrical impulse? Again, it's the flip of the membrane polarity. You are changing the membrane polarity. Rather than inside to be negatively charged, inside will become positively charged, more positively charged than the outside, by allowing what ion, again, what ion is responsible for the depolarization of the cell is my sodium, exactly. And in order for me to conduct another signal, you can't conduct a signal while you have a signal already in the cell. So you can't depolarize while you are depolarized. You need to go back to the polarized state for you to depolarize another time. So what do we call the process by which we're going to be returning back to the polarized state? This is my repolarization. And something to remind you here, the ion responsible for the polarized state is going to be my potassium. So in order for me to get back to the polarized state, I will allow what ion to travel? Again, my potassium. All right. So the efflux of potassium to the outside of the cell is the one that's going to be responsible for you to regain your polarized state. All right, so I don't want to give you more information today. I want you to read this part from your slides until we meet on Thursday. Also, make sure you dedicate enough time to prepare for the muscles lab exam. You have only one attempt to complete the muscles lab exam. So please make sure you did prepare well enough before you take the exam. All right. So we're gonna discuss more about the action potential and the electrical conduction through the nerves. This is just a brief introduction. This is not the whole topic yet. We're gonna keep going. I will review this at the beginning of next class, and I will keep going to cover the rest of the topic. Again, one of the most important topics is this semester, and you're gonna rely on this topic in order for you to understand EKG, in order to understand the cardiac physiology, and a lot of other topics that you're gonna be learning more about in AP2. So it's a very, very important topic. And again, this is gonna be an essay questions that you're gonna have on the last lab exam. I'll tell you the format exactly of the essay question for you to prepare for it. But this, everything that we've mentioned so far is gonna be part of this essay question, of your answer for this to the essay question. Any questions? Any questions? All right, thank you for making it all the way till the end of the class. And we'll see you on Thursday. Have a good rest of your week. Have a good night and stay safe. Thank you so much.